Welcome. My name is Åsa Hidmark. I'm an immunologist from Sweden. I'm working in Heidelberg. I have made 3 lectures for the students on the infectious disease program in Heidelberg University. I have now divided these lectures up for YouTube and adapted them a little bit so that you can also take part of these lectures. The first lecture series is a lecture on mucosal defenses. It is partly innate immunology, or innate immunity to infectious disease on mucosal surfaces, but it's also a general description of the mucosal surfaces. I think this is particularly interesting right now when we have a discussion about gut leakage, tight junctions, and also the lung physiology and the mucosal surfaces in the lung. So this is what you will get in this mucosal lecture. Then there are 2 other lectures, which is innate immunity and vaccine. And I will try to adapt both of them for YouTube also, but we will see when they come.
This lecture is divided up in 3 parts. First, we will talk about general features of the mucosal surfaces with a special focus of epithelial cell function. Thereafter, we will move on to the intestinal tract, where we'll talk about the intestinal surfaces. And we will finally talk about respiratory tract. So, to first define the mucosal surfaces, or mucosa, or Schleimhaut, German, it's the membrane lining of various cavities in the body. It has 1 or more layers of epithelial cells, and they are connected by connective tissue. Many of the mucosal membranes, they secrete mucus, and mucus we will come back to because it's very important. Mucosal surfaces are found in the mammary gland and in the urogenital tract, which is the bladder, the kidneys, and the vagina. The uterus also has a particular type of epithelial cells, but we will not focus on these parts. We will rather focus on the respiratory tract and the lungs, and on the digestive system, and most important, the guts. There are several different types of epithelium, and some of them are in particular important to the subjects of today. The first is the simple squamous epithelium. This can be found inside of the respiratory spaces in the lung, which is the alveoli. The second type, which is important for this lecture, is the simple columnar. We find this in the small intestine and in the colon. This layer of epithelium is much thicker than the simple squamous one, and it's specialized in absorption and in secretion of mucus. The third type, which is important to us, is the pseudostratified. This is found in the trachea and the bronchial tree. Here we see also a lot of secretion. So the epithelial barrier is protecting us mainly through 3 different pathways. It's a physical barrier to infection, and tight junctions between the cell types in the epithelium is important here, and we'll come back to that. The mucus production we will come back to, and the chemical defense. The second function of the epithelial barrier is the killing of microbes by locally produced antibiotics, defensins and cathelicidins, and these are the antimicrobial peptides, which we will talk about. The third part is the innate immune system, which we will also talk much more about tomorrow. Here, the intraepithelial lymphocytes will kill both microbes and infected cells, and they have several different subtypes and classes and functions. So, part of the protection against infection is the non-specific physical barriers, and epithelial cells constitute some of these. Here, most of all, we are going to talk about the mucus and the cilia, which is both, the cilia is in the airway, and the mucus we find both in the airway and in the gut. And we are going to talk about the commensals in the gut. But first we are going to talk about the tight junctions and the bacterial peptides, which are produced by the epithelial layer. So first we will talk about the physical barrier to infection. I will talk about tight junctions between cells in the epithelium, and we will talk about mucus production when we talk about goblet cells. So the physical barrier is constituting a very large area, over 400 square meters, and it's a multicellular sheet where the cells are held together by the tight junctions. The tight junctions are also known as occluding junctions, or in Latin zonulae occludentes, and they are multi-protein junctional complexes that functions in order to prevent leakage of water out from the body. The proteins bridging the cell and connecting to the other cell is constituted by 3 different types of proteins. It's the occludins, it's the claudins, and the jam protein. While the claudins and the occludins have a similar structure with several transmembrane regions, the jam proteins are part of the immunoglobulin superfamily, and it only has one single membrane spanning region. On the inside of the tight junctions, there are several different proteins that are anchoring the tight junctions to the actin filaments in order to keep the junctions stable. These proteins are called zonulins, and they will, in the gut, regulate the permeability between the cells. The tight junctions in the gut are found just below the mucosal surface, at the apical side, which is towards the outside of the body. So in addition to the tight junctions, there are also 2 other cell-to-cell contact manners which are worth mentioning when it comes to epithelial cells. The zonula occludens is the tight junction, and the zonula adherens is also present on endothelial and epithelial cells. It is a band that is going around the cells, and the cell-to-cell contact is mediated by E-cadherin rather than the claudins and the occludins. Just like the tight junctions, the intermediate junctions also attach to the actin filaments. However, the desmosome is the type of junction that does attach to the intermediate filaments. The desmosome is a spot junction. It has 1 point of adherence, which is randomly distributed between the cells. These kinds of junctions are found in cardiac muscle, but also in bladder tissue and gastrointestinal mucosa. The desmosome is a work of cadherin proteins, linker proteins, and creatine intermediate filaments. So the second part is the production of antimicrobial peptides as a way of direct killing of bacteria. The chemical barrier, which is of particular importance for the immune system, is the antimicrobial peptides. There are basically 2 types of antimicrobial peptides divided up on their structures. The catholycidins are alpha helical and the defensins are beta sheet formed. There are also mixed versions. What is common for all the antimicrobial peptides is that they have at least 2 positively charged residues, and they are more than 50% hydrophobic. That means that one side, which is hydrophilic, will be attracted to the negatively charged phospholipids in the membrane, and they will embed there. The hydrophobic part of the antimicrobial peptides will embed into the lipid bilayer, where it will cause damage to the membranes. The damage to the membranes can be a general type of destabilizing or actual pore formation. The antimicrobial peptides also have immunological functions, for example, inhibiting polysaccharides-induced pro-inflammatory cytokine production, and antimicrobial peptides will promote wound healing. On the mucosal surfaces, the antimicrobial peptides are important for keeping a zone very close to the epithelial cells clear from bacteria. In this lecture, we will come back to discussing how the production of the antimicrobial peptides is regulated in the epithelial cells. In this slide, I show the mechanism that I described previously, how the defensins will enter into the membrane by being attracted to the phospholipid head of the membrane of the bacteria. Thereafter, it will insert its hydrophobic part inside of the bilipid membrane, and there the pore formation will happen. And there is a small GIF film here, and I hope it works. So, talking about defensins, they are very active in regulating immune responses as well as acting on the membrane of bacteria. They function as chemoattractants for T lymphocytes, and they can dampen the expression of pro-inflammatory genes in macrophages. As mentioned before, they can inhibit the LPS-induced cytokines. They will also induce the expression of co-stimulatory molecules. They will enhance the phagocytosis by macrophages, and it will increase, the defensin will increase pro-inflammatory cytokine production. There are alpha defensins and beta defensins. The beta defensins are cationic, and they are found in the tongue and skin and cornea and kidneys. And toll-like receptors are inducing the expression of the beta defensins. The alpha defensins are mainly produced in the Paneth cells, which we'll come back to. They reside in the granules of neutrophils and macrophages, so it's more of a immune cell type produced defensin. We found them in the respiratory tract, the urinary tract, and the vagina. The cathelicidins is the alpha helical formed antimicrobial peptide, and the most notable is the LL37. The LL37 expression is induced by vitamin D, and the cathelicidins will destroy the lipoprotein membranes of microbes inside of the phagosomes after fusion with lysosomes. So it's not actually secreted. It is produced by the epithelial cells themselves.